Hi, physiology students, and welcome to chapter 12. This chapter is all on muscles, specifically skeletal muscles we spend a lot of time with, but we'll review cardiac and smooth muscle at the end. Some of this will be a review, so I will skip over some of the things that are a review, and I'll let you know if you need to know those. I may skip over other slides as well that you don't need to know, but I'll try to make that very clear. Generally, both ends of a skeletal muscle are attached to a bone by tough tendons, and when the muscle contracts, it shortens. And this shortening will move the bone at the joint. The bone that does not move is attached at the muscle origin, and the movement is toward the insertion. So origin and insertion of muscles, different movements depend on the joint and how the muscles are attached. So we have flexor muscles decreasing the angle, extensor muscles increase the angle. The main muscle responsible for movement is the agonist or the prime mover, and flexors and extensors that act on the same joint to produce opposite actions are antagonists. And that would be like your biceps and triceps. Your biceps flexes your arm, your triceps extends the arm. I hope some of that is a little new from anatomy. You should have a good understanding of these categories of muscles and what their action is. You probably learned this in anatomy, but I'm going to have you learn it here in physiology too, because in your healthcare related fields, you might read notes on a patient that is listing these actions of their joints. Connective tissue components, I'm going to skip over this. This takes you through the structure of skeletal muscles. I don't know if I'll ask you too many questions about the structure, but you should generally remember that a skeletal muscle is made up of fascicles, and fascicles are made up of muscle fibers, which are the muscle cells. Muscle fibers are made of myofibrils, and the myofibrils are made of myofilaments. And within these myofilaments is where we'll focus, and that's where contraction occurs. Muscle fibers are striated. The I bands are light bands, the A bands are dark bands, and the Z lines are the discs that are the dark lines in the middle of the I band. So this is what skeletal muscle looks like. You can see it's striated. The nuclei are pushed off to the periphery. Motor unit. A motor unit is a single motor neuron or nerve cell and all the muscle fibers it innervates. All the muscle fibers in a motor unit contract at once. We can have graded contractions, which are varied contraction strength due to a different number of motor units being stimulated. The neuromuscular junction is the site where a motor neuron will stimulate a muscle fiber, and the motor end plate is the area of the muscle fiber sarcolemma or membrane where a motor neuron stimulates it using the neurotransmitter acetylcholine. So this is a look at a neuromuscular junction and the motor end plate. You should be familiar with this. So these are branches of the motor neuron coming out. Here's the folded sarcolemma, which contains kind of this branch of the motor neuron. You have synaptic vesicles containing acetylcholine, which will be released across the synaptic cleft and bind to receptors on the motor end plate. So that is how we stimulate a muscle. Here is a look at a motor unit. So again, a motor unit contains the motor neuron and all of the nerve muscle cells that it innervates. So this is one motor unit from this somatic motor neuron and another motor unit from this somatic motor neuron. Control of motor units, contraction strength comes from motor unit recruitment. Finer muscle control requires smaller motor unit. So the eye muscles may have about 23 muscle fibers per motor units, whereas larger, strongest muscles like your quadriceps muscles may have motor units with thousands of muscle fibers. Control and strength are trade-offs. So you'll notice your leg muscles are extremely strong, but they're not as precise as doing fine motor movements like your fingers are. Duchenne muscular dystrophy accounts for half of more than 30 genetically different muscular dystrophies. It's caused by a mutation in a gene located on the X chromosome having to do with a protein called dystrophin. Basically, the mutation just causes the sarcolemma. It cannot be repaired by satellite cells, and it just pretty much causes that muscle fiber to die off, and the replacement is by just fibrous connective tissue and fatty tissue instead of more muscle fiber cells. So the mechanism of muscle contraction you should be well familiar with. You should know how each fiber looks. It's densely packed, called myofibrils. It runs the lengths of the muscle fiber, and it's composed of thick and thin myofilaments. So this is a look at a muscle fiber cell. Again, a muscle fiber is a muscle cell, so you can see the nucleus. The myofibrils make up the muscle fiber, and within the myofibrils are these myofilaments, which will perform the contraction or shortening of the muscle. The striations are produced by thick and thin filaments. Here are the I bands, the A bands, the H bands, and the Z discs that you guys can review on your own. I will show you a picture right now, and the previous slide describes what is in each, but here's your H band, includes the myosin-thick filaments. The Z disc is where the actin, the thin filaments, attach to. You can see here what makes up the A band, where the myosin and actin overlap, and the I band is only actin filaments. The sarcomere is the basic subunit of striated muscle contraction. It goes from one Z disc to the next. My M lines are found in the center of each A band and help hold down the thick filaments. So that is the sarcomere. And again, you can see these bands are labeled and how the Z disc kind of holds down the thin filaments. And then the thick and thin filaments, which are found here in the A band, will slide past each other and shorten in order to contract the muscle. And this is a great look. Again, pause the video, just take a look at where these bands are located, and then go to that previous slide to learn what's involved in each of the bands. Tightened filaments. So a tightened filament is a specific type of protein that anchors to the thick filaments and the M lines. The M line goes right down the thick filaments. When a muscle contracts, the sarcomeres will shorten. The A bands will not shorten, but they'll move closer together. The I bands do shorten. In general, when you're talking about muscle contraction, your thin and your thick filaments never shorten. They just slide past each other, so the area where their overlap shortens. So this is a look at the sliding filament theory, showing in the sliding filament theory, it describes muscle contraction. Here's a relaxed muscle, and you can see how partially contracted and fully contracted, it basically shortens the sarcomere because the thick and thin filaments here shown with the thin line and the thicker green line are just sliding past each other. Here's a summary of the sliding filament theory of contraction. And this is a great chart to study, and I would pull a lot of information from the tests on this. So you should read through this. Really simply put, it talks about, I'm going to skip to number 3, the shortening of the sarcomeres is accomplished by sliding of myofilaments. Sliding of the filaments is produced by asynchronous power strokes of myosin cross bridges. That means that myosin will form a bridge or an attachment with the thin filaments, and it will pull the thin filaments over the thick filaments. So that is what the sliding filament theory is. Your thick filaments are forming a cross bridge or attachment with your thin filaments. They're sliding past each other to perform a muscle contraction. So cross bridges are formed. Thick filaments are formed of the protein myosin, and these myosin has 2 globular heads. It kind of looks like a golf club with actin binding sites and ATP binding sites. Your thin filaments are composed of the protein actin, and they have proteins called tropomyosin and troponin that will prevent myosin binding when the muscle is at rest. So your thick and thin filaments can only bind together when the muscle is being contracted and when calcium is available. And we'll talk about how that is. So here we have here's your thin filament. It looks kind of like a double wound strand of beads. The troponin is this large protein-like structure, and then this tropomyosin are these spaghetti-like proteins. The tropomyosin cover up all of the binding sites that the myosin heads will bind to, and the troponin will be proteins that bind to calcium. When calcium binds to troponin, that will slide tropomyosin off of these binding sites, so that these thick filament, these myosin heads, can reach up and form a binding or connection with the thin filament. When that happens, you can see here this myosin head, the hinge segment. It's basically ready to kind of do the power stroke, which is the pulling of the thin filaments past it. In order to do that, the ATP will hydrolyze or break apart, and then the power stroke occurs. So you can see here that it's kind of in this straight angle. The action of sliding, it's produced by several cross bridges that form between the myosin actin. The myosin head serves as a myosin ATP ACE enzyme. It splits the ATP apart, and this allows the head to bind to actin when the muscle is stimulated. So this release of the phosphate group upon binding will cock the myosin head, producing a power stroke that pulls the thin filament towards the center, and it slides the thin past the thick filaments. After the power stroke, ADP is released and a new ATP binds. This makes myosin release actin. ATP is split. The myosin head straightens out and rebinds to actin farther back, and it continues until the sarcomere has shortened. So this takes you through kind of the cross bridge cycle. An important thing to note is after the thick filament has been bound to actin, in order to kind of release the attachment, more ATP is required. So you actually need energy to release the thick filament head from the actin. This is contraction, regulation of contraction, and it kind of takes you through the troponin complex. The tropomyosin were those spaghetti-like strands that physically block the cross bridges. The troponin is made up of different kind of complexes, but really simply put, the troponin complex, you don't need to know, there are 3 kind of subunits to it, but all you need to know is that when calcium binds to troponin, it changes the shape and moves the tropomyosin off of the active sites where the myosin head can bind. So when a muscle cell is stimulated, calcium is released from inside the muscle fiber, and that calcium is what will bind to troponin, causing a conformational change and move tropomyosin off of the binding site so that myosin is allowed to form a cross bridge with actin. And you can see that role of calcium here. The sarcoplasmic reticulum, it stores calcium when the muscle is at rest, and when a muscle fiber is stimulated, calcium gets released from the sarcoplasmic reticulum. At the end of contraction, calcium will be actively pumped back into the sarcoplasmic reticulum for further use. So you can see here the sarcolemma is like the plasma membrane of the fiber muscle cell. The sarcoplasmic reticulum is seen in green here, and an electrical stimulus travels down these T tubules, and then that electrical stimulus will cause the calcium to be released from the sarcoplasmic reticulum, and that calcium is what you need for muscle contraction. The transverse tubules, these are these narrow tunnels. They're formed from the sarcolemma, and they are able to conduct action potentials or an electrical signal down into the muscle fiber. They're situated next to the terminal cisternae, and from those terminal cisternae, that's where your calcium will be released from. So this is a look at a somatic motor neuron, how an action potential travels into the sarcolemma, down the transverse tubules. From there, the action potential will open voltage-gated calcium channels, which will allow calcium to be released from the sarcoplasmic reticulum, and then calcium will then bind to troponin, stimulating that contraction. So here's a look. This is a great picture describing these steps and kind of what happens, but here's the neuron sending out its neurotransmitter to stimulate the muscle. So acetylcholine binds to receptors at 1, and then that causes an action potential to travel down the sarcolemma and eventually down the T tubules into the muscle fiber. This will cause different channels to open, and eventually it will cause calcium to be released from the sarcoplasmic reticulum. And again, we need calcium in order to bind to troponin, move the tropomyosin off the binding sites so that myosin heads can bind to actin. So what happens when muscles relax? The action potentials will stop, meaning there's no more stimulus going to that muscle. Calcium release channels close, and calcium will be pumped back into the sarcoplasmic reticulum. Because no more calcium is available to bind to troponin, the tropomyosin moves back to block all of those myosin heads from binding to actin. So in terms of skeletal muscle contraction, we can do a study done in a lab where one end of the muscle is fixed and the other is movable. Electrical stimulations are applied and the contractions are recorded and displayed as currents. A twitch is when a muscle quickly contracts and relaxes after a single electrical shock, and increasing the voltage increases the strength of a twitch up to a maximum. If a second shock is applied immediately after the first, the second twitch will partially piggyback off the first, and this is called summation. The latent period is the time between the stimulus and the contraction, and a graded contraction means that the stronger the contractions will result in recruitment of more fibers until all fibers are contracting. So this just shows an example done in lab showing how muscles will twitch or respond to a stimulus. So here's a stimulus and showing a muscle twitch. Here are two stimuli kind of right after each other, and they are done before the first muscle twitch can relax all the way. So these muscle twitches will summate or become larger. Tetanus refers to increasing the frequency of electrical shocks, decreases relaxation time between the twitches. This is called incomplete tetanus, whereas at a certain frequency, there will be no relaxation. And this is called complete tetanus, which is a smooth, sustained contraction. And this is shown here in these pictures in the graph. Tetanus in vivo, this means in real life. This is asynchronization activation of motor units, which means that some motor units will start to twitch when others start to relax. And this produces a continuous contraction of a whole muscle. Recruitment will make these contractions stronger. Trepe refers to the number of muscle fibers used in vitro increasing as voltage is increased. This will reach a maximum value when all muscle fibers are stimulated. If a fresh muscle is stimulated with several shocks at maximum voltage, each twitch will be progressively stronger. And recorded, this produces what we call a staircase effect called trepe. You can skip the force velocity curve. An isotonic contraction is when muscle fibers shorten when the tension produced is just greater than the load. So a muscle fiber shortens when the force is greater than the load. That's a concentric contraction. And an eccentric contraction refers to a muscle may actually lengthen despite the contraction if the load is too great. An isometric contraction are where muscles can't shorten because the load is too great. This can be voluntary. So those are the two types of contraction. Series elastic component, it talks about tendons being elastic to try to resist distension and snap back to their resting length. You don't need to know about the length-tension relationship, so I'm going to skip over that for now. Energy requirements of skeletal muscle. So basically, all I want you to know is that for energy to contract, it needs ATP and calcium. And I'm going to kind of skip over the rest of this. Well, a couple things you should know then. Sorry, that you can get energy from glycogen stores for moderate exercise, and for heavy exercise, you can get it from blood glucose. Metabolism of skeletal muscle, I'm going to skip over for now. It talks about requiring oxygen, and you'll get to lactic acid buildup. That's the burn you feel if you do too much exercise. But I want to talk a little bit about slow and fast twitch fibers. Slow twitch are slower contracting speeds. They can sustain contraction for long periods without fatigue. They have a rich blood vessel supply, lots of mitochondria, more respiratory enzymes, and more myoglobin. They are said to have high oxidative capacity, so are called slow oxidative fibers. They're due to a high myoglobin content, which has a red pigment, so they're also called red fibers because they have a red pigment to them. These are found in postural muscles, We also call this the dark meat in poultry animals that we eat. So think about the turkey, the bird, their dark meat is found in the wings and the legs, the area of slow twitch, slower contractions. These are muscles that need to perform contractions for longer periods without fatiguing. So found in your postural muscles in the back, but also in legs of a kind of a consistent organism that would be doing a lot of running. Fast twitch, faster contraction speeds, they'll fatigue faster, fewer capillaries, mitochondria, respiratory enzymes, less myoglobin. They're called white fibers, so we sometimes call them the white meat, and they're found in stronger muscles that might just need kind of a burst of energy. You have intermediate fibers as well. People vary greatly in the percentage of fast or slow twitch fibers in their muscles, and this can be a result of genetics or training. So if you train for a marathon, you'll probably develop a lot of slow twitch muscles. If you're a bodybuilder who's trained to just do reps really quickly of a high weight, you'll have more fast twitch muscles. So this is a great chart to kind of just review the slow and fast twitch muscles and what are certain characteristics of both. So you should have a basic understanding of this chart. This shows you this is an interesting chart to look at where their abundance of certain people and where their fiber types would lie. So here is an extreme endurance athlete. Think of someone who does triathlons, marathons. They have a lot of slow type muscles because they can be constantly contracting without fatiguing. World-class marathon runners, middle distance runner, a person with a spinal injury will have hardly any slow type fibers. They just have the fast fibers. Muscle fatigue is due to basically accumulation of extracellular potassium, which will reduce the ability to be stimulated, reduce sarcoplasmic reticulum calcium release. I'm going to kind of go ahead. Adaptation to endurance training is very interesting to read about, but I'm going to skip ahead this for now. Muscle decline with aging. I do want you to know with aging, your muscles will reduce their mass. It can be helped with strength training. So as you age, I'd encourage you guys to lift weights every day for a couple minutes. There's a reduction in capillary blood supply. It can be helped with endurance training as well. So basically what it's telling you is exercise is really important, whether it's walking or weightlifting as you age. Muscle damage and repair, it's harder for cells to repair as you age as well. Adaptation to strength training, your type 2 muscle fibers will become thicker. They will hypertrophy. Atrophy is a reduction in size with its accompanying declines in strength. It's most severe when it results from damage to motor nerves innervating the muscles, because if your motor nerves are damaged, they won't be able to stimulate these muscles, so your muscles won't be able to contract and be able to gain size. Usually this has to do with diseases like ALS or other trauma. Disuse atrophy can result when a person is bedridden or is in a cast, so the muscles will not be working as they normally do to move a load against the force of gravity. So you'll notice if you break your leg or your arm, the one that was broken, that was in the cast, will be atrophied. It will have a smaller size to it. Neural control of muscles. I'm going to skip a little bit over this for now. Lower motor neurons and upper motor neurons, muscle sensory organs. I want to get to the muscle spindle apparatus. This will just innervate muscle spindles will sense any change in the muscle. So it's a sort of a sensory response that your muscles have. I'm going to skip ahead a little bit to reflexes though. I want you to know a little bit about reflexes. Skeletal muscles are usually referred to as voluntary and are controlled by descending motor pathways, but they can also contract unconsciously without you knowing they are in response to a certain stimuli, and we call this a reflex. A monosynaptic stretch reflex is the simplest reflex. It only involves a sensory neuron synapsing on a motor neuron. It will maintain optimal resting length of skeletal muscles. And this is the knee-jerk reflex that your doctor tests when he strikes your patellar ligament with his rubber hammer. He's testing your monosynaptic stretch reflex. These are the events in a monosynaptic stretch reflex that you can read. And here's a look at the monosynaptic. It's called the knee-jerk reflex. So just understanding this is an example of a one synaptic reflex, meaning when your doctor hits the patellar ligament, the muscle spindles, which are sensory neurons, will take that sensation right to your spinal cord. It'll synapse on one motor neuron, and the motor neuron will go right to your quadriceps muscle, and it'll kick your leg up instantaneously. This happens instantaneously, and it's called a monosynaptic because there's only one synapse or changeover of signal from a sensory to a motor neuron. Golgi tendon organs, we can skip over that. I want to talk a little bit about the crossed extensor reflex. So the crossed extensor reflex is a type of double reflex, reciprocal innervation seen when you step on a tack. And it's called the crossed extensor reflex because what it'll happen when you step on the tack, it'll send a sensory information to the spinal cord to basically tell you the flexor contracts and the extensor relaxes to withdraw the foot from the pain. But also on the opposite side of the body, the extensor contracts and the flexor relaxes in the other leg to support your weight. So simultaneously, if your right foot steps on a nail or some sort of painful stimulus, that stimulus causes your left leg to counteract the balance so that you won't fall over. So that's called the crossed extensor reflex. It actually tells the opposite leg to account for any balance makeup if you have to lift your leg off that painful stimuli. And I'm going to skip over this, the upper motor neuron control. Types of paralysis, damage to the lower motor neurons or spinal nerve is called flaccid paralysis. And this will involve reduced muscle tone, depressed stretch reflexes, atrophy. Damage to the upper motor neurons or descending tracts is called spastic paralysis. This will cause increased muscle tone, exaggerated stretch reflexes, and hyperactivity. You don't need to know these symptoms. ALS is a neurodegenerative disease that affects both lower motor neurons in the spinal cord, leading to muscle wasting, and also upper motor neurons, leading to scarring or sclerosis of the muscles. It will basically produce progressive muscle weakness. Death usually occurs from respiratory failure because your diaphragm is a muscle, and if that fails, you're unable to breathe. Cardiac and smooth muscle. So this is just where we'll end the PowerPoint, reviewing cardiac and smooth muscle. This should be a review from anatomy, so I'll let you guys review this on your own, kind of what the characteristics of cardiac muscle are versus smooth muscle, what they both look like. So here's cardiac muscle connected. The intercalated discs have gap junctions, which just allow the electrical signal to pass through simultaneously. Cardiac muscle is involuntary, striated. The myocardium is the mass of cardiac muscle cells connected to each other. It behaves as a single functional unit. And basically, you don't need to know the role of calcium channels in cardiac muscle. You should just know kind of in general what cardiac and smooth muscle are, where smooth muscle is found. No sarcomeres, it still contains a large amounts of actin and myosin. Its arrangement allows contraction even when greatly stretched. So no striations, also involuntary. There's single unit and multi-unit smooth muscles. You don't really need to know that either. But again, just knowing the basic kind of differences between smooth and cardiac muscle is all I'm going to ask you to know about that. Don't worry about excitation, contraction in smooth muscle. I just want you to understand that in skeletal muscle. And this is probably actually a great chart to look at the differences between skeletal, cardiac, and smooth in terms of reviewing them. And that's the end of this chapter. Thanks for listening, guys.